So it's the cards were not right.
Yes. Ask for card.
You get it. Also, right, yeah, it's the same. You have it.
Non, non, vas-y.
C'est vrai qu'il y en a qui
Second,
so what you said about three was very last.
Hello everyone, and welcome to this back seminar for the autumn holidays, where we will have a 2 weeks break. And so, as usual, this is a seminar in webinar mode. So there are people online, so we can't interrupt right now, but we will be also authorized to ask questions. So at the end of the seminar, we will have a question session, both with people from the room and people online. And I'll let you introduce Natacha.
Yeah. So we are really pleased to welcome Natacha today for this seminar. I'm going to present really quickly what she did, her work, but of course she will present us more detail. So she started in Lyon with Fabrice Fabre on a PhD on the evolution of dependency in Wolbachia symbiosis. And you will see that symbiosis is sort of the main topic of her work, basically. And then she went to Wisconsin University to work with Margaret McFall on the mutualistic association between the bacteria Vibrio fischeri and the Sepiula Euprima scrophulales, focusing on the molecular dialogue happening during the establishment and the maintenance of the symbiosis. And then Natacha get a position in the genetic and evolution of interaction team at the CNRS in Lyon. And she developed multiple research projects focused on symbiosis and the evolution of interactions between the insects host and, of course, the gathering of microorganisms. And I guess you will present that to us. So thank you very much. And yeah, process yours.
Thank you very much for the introduction. Thank you, Sido, Nicola, and Cindy for the invitation. I'm really happy to be here today. And what I'm going to do is not to focus on one particular project in details, but rather give you a general overview of what I did. And in particular, it's a question that I asked together with Anna Rivero during the Perspective du CNRS that was about considering the host as an ecosystem. And so today I'm going to build a little bit on this topic. So as Sido mentioned, I mostly work on symbiosis. And so for me, symbiosis is a long-term interaction between two partners, a big one and a small one. So the big one is a host and the small one is a symbiont that can be another symbiont. And what is interesting to me is more the balance between the costs and benefits that is associated to this partnership. So the presence of symbiont can be costly for the host because it uses the host niche machinery or the nutrients that are present in the cell. It can induce immune defenses. And sometimes some symbionts can also provide benefits, such as provision of nutrients, protection against pathogens, or stress tolerance. And so based on the balance between these costs and benefits, you can put the association along a gradient, a continuum between parasitism and mutualism. And this position can move across time and can also move depending on the environment that is changing around. And this week I was listening to a conference by Charlotte Brice that mentioned the term of pluribiosis. I never heard about this term before, but it's an interesting case in which she considered pluribiosis as different snapshots of this interaction during the lifetime of the organism. And so what is important in symbiotic association is the transmission mode. So classically, we talk about vertical transmission when symbionts are transmitted from mother to offspring through the eggs mainly. And so the offspring will have exactly or almost exactly the same composition than the mother. And in that case, you observe a general alignment of interest between the partners, and it leads to the evolution toward cooperation and mutualism, but it doesn't mean that there is no conflict at all in the system, but the main outcome is in general mutualism. In horizontally transmitted symbiosis, on the opposite, the offspring will acquire symbionts from the environment, and so the composition of the symbiotic community can be very different from the one of their mother. And in this case, the interest between partners is less aligned, and we generally have an evolution toward conflict and parasitism. But again, this is the main rule, but sometimes you can have cooperation, especially when there's partner choice, and I will show you an example later on. So now I'm going to tell you a little bit about how we classically work on symbiosis and how this field is changing over time. So in general, what we were used to do is to look at one host and one symbiont. So what I mean by symbiont is all this continuum between parasitism and mutualism. And so looking at this specific interaction, what we can do is to look at the impact of symbiont on host life history traits, the impact of the host on the symbiotic dynamics, and the mechanism of interaction and the molecular dialogue, because since you have only two partners, it's easier to disentangle who is doing what. And then we took into account the fact that there can be variation within the host and within symbionts, so you have different genotypes. And if you consider the different genotypes of host and symbionts, then you can address the question of within host and between host competition, evolution of virulence and cooperation, evolution of resistance, tolerance, and mechanisms of harm price or coevolution. And more recently, and the environment obviously can change the game. And more recently, we started to consider one host and multiple symbionts within a host, and with different genotypes. And what is important here is that each symbiont can potentially modify the host extended phenotype. And so if you consider multiple symbionts within a host, then you will have multi-party interactions, and you can study competition, facilitation, exclusion between different symbionts. And you can also apply more general concept on community ecology, such as the establishment of communities, stability, regulation, and dysbiosis. And so, and again, the environment can be integrated in this framework. And so what I would like to talk today is to consider the host more generally as an ecosystem. And so the ecosystem has been defined by Odom in 1953 as the largest functional unit in ecology. It includes both living organisms and their biotic environment, each influencing the properties of the others. And so if we consider the host as a symbiotic ecosystem, then we can say that the living organisms are the symbionts. We have an abiotic environment around the host, and all of these influence the properties of the other. And that's what I'm going to present today. I focus on 4 major questions that we're interested in, that I will develop point by point. And I will mainly focus on my past project on the squid and on Drosophila. And if I have time, but I'm not sure because I'm a bit chatty, I will present the current project I have on bedbugs. So if we talk about the assembly rules in each ecosystem, you have some assemblies. And here I will only focus on horizontally transmitted symbiosis, because in general you have a complex community around, and only part of it will be integrated within the host. And so we have specificity, and there's a process of filtering that occurs here, and I was wondering about which mechanism could play a role in this filtering. And more generally, I would like to understand what could be the influence of the orthogonal feature and host and symbiont genotype in this composition, the influence of diet, is there nutrients that promote the growth of certain symbionts and not others? Is there an influence of the host niche during the colonization process? Is there a kind of differential survival during the colonization, depending on the organ where they are located? And is there an influence of other symbionts that are already present in the host, vertically transmitted symbionts or already established communities? So first, I will give you an example on the squid bubble system. So what is this system? You may have heard about it before. It's so here you have a little squid on the left, Euprymna scolopis. So when they hatch in the seawater, they are ecosymbiotic, so they don't contain any symbionts, and they will acquire Vibrio fischeri from the seawater. And what is interesting is Vibrio fischeri represent only less than 0.5% of the bacterial plankton. So, but it's the only one that is able to colonize the light organ. So there is very strong specificity. And so it will colonize. So this is the light organ. In fact, what happens is that the bacteria stick onto the appendages of the light organ, and then they will move from the appendage to the pores, and then they will enter into the pores and migrate to the crypts that are inner structure within the light organ. And so the question I was asking here are what mechanisms are involved in this filtering? Do we have a selective incorporation? So we have sorting at the beginning. Selective accommodation, so everybody gets in, but the squid will only select a few for survival. Or is there any selective maintenance? And so everybody gets in, but the ones that are not producing light or that don't provide any benefit are sanctioned. And so what we did first is to look at how Vibrio fischeri bacteria can be recognized. So we saw that this bacteria can release vesicle-like structure that contains LPS or lipopolysaccharides that are highly reactogenic and that have a very specific signal compared to the other bacterium from the seawater. And we look on the microscopy and we could see that only 5 cells attached to the epithelium of the cilia. So it's very few cells. And what I was interested in is determine if the contact with these few cells could change the gene expression in the light organ, and if the squid would respond to a contact with very, very few cells. And indeed, there are a set of genes that are differentially expressed in response to this first contact. And we went a little more in the characterization of these genes. And we could find the hemocyanin and lysozyme that are proteins that are antimicrobial and that kind of create an antimicrobial cocktail in which only Vibrio fischeri can survive. So there are other proteins as well, but these ones are important. We also find a chitotriosidase. So this is an enzyme that can create chemoattractant gradients of sugar. And so this gradient can be followed by Vibrio fisheye when it's prime, and it helps Vibrio fisheye to go from the inner to the inner part of the pit. And there is also cathepsin L, which is a component that induces apoptosis, and which is associated with the regression of the appendage. I will talk a little bit about that after. But this is a way to kind of close the door when Vibrio fisheye is already in. And so all these results suggest that it's mostly a selective incorporation of the symbiont in this system. Another example, which is much less detailed, it's on symbiotic community in Drosophila. It's a project that is still going on. So what we know in Drosophila is that they acquired horizontally transmitted symbiont, especially in the gut, and they have a gut microbiota. And in Drosophila melanogaster, there are two main genus that are present in the microbiota, which are Lactobacillus and Acetobacter. And these bacteria can have a strong impact on nutrition, immunity, and communication, and they are localized in gut tissues. And there's also in some population a vertically transmitted symbiont, which is called Wolbachia. It's an intracellular bacterium that in melanogaster induces very weak reproductive manipulation. And it's also localized in ovaries and in all tissues, almost in Drosophila. And so the question, we know that in the adults of flies, there is an influence of the diet and genotype on the composition of this microbiota, but it doesn't seem to be a core microbiota if we speak in terms of taxon. And so the question we ask is, in the larvae, what is the composition of the gut microbiota? Is it very different from the host, from the adult? And mainly because larvae is a stage in which they are only eating, mostly. And so they can acquire a lot of bacteria during this phase. So that's the reason why we were interested in this stage. And we try to determine which factors can shape the community composition in larvae in terms of diet, location, and the presence of other bacteria like Wolbachia. So it's a project we did with the LabX EcoFect with François Lelier and the postdoc Vincent Raquet. So what we did is we sampled different places fruit flies using banana or cherries. And we identified the ones that were melanogaster by RFLP method. We collected the decaying fruits and the larvae. We dissected the gut, and then we did 16S sequencing for the bacterial composition and ITS2 sequencing for the fungal composition. And we quantified the quantity of Wolbachia. So what we could see is that, not surprisingly, the bacterial community in fresh fruit is very different from the one from rotten fruits. But more interestingly, the community in rotten fruits, which is the one that larvae eat, are very different from the one that we find in the guts. So there is a process of filtering between what they directly eat and what we can find in the gut. We could see an influence of the bait and of the collection site on the structure of this community, but the presence of Wolbachia didn't seem to impact any composition structure. So we need to go a little more on these results, but overall it's quite complicated, especially on the fungal part, because there is a limited resolution in terms of taxonomy, and so we need to develop more detailed fungal databases. We would like to go a little more in the timing of colonization, so which species are pioneer species, keystone species, and how stable are these communities. And for that, we need to consider the spatial and temporal dynamics and the interaction between microbes within organs or tissues. We can also integrate community ecology tools in this type of system or manipulate the system by introducing sequentially different symbionts and to see if we can reproduce the normal colonization. And one step further would be to look at the impact of this community composition on the host physiology or immunity. And instead of using a taxonomic scale, rather considering the function and the metabolites that are expressed within each bacteria or fungi to better understand their role in the association. The second point I would like to address today is the control and maintenance of these symbiotic communities. So as I showed you before, there is a balance between costs and benefits in the host, which is oftentimes linked to the density of symbionts. And on parallel, there is also, from the symbiote side, another trade-off between transmission and virulence, which is also oftentimes linked to density. And for these reasons, most of the time we get to an optimum of density that maximizes the fitness of both partners. And if there's an optimum, it means that our control mechanisms, either in terms of immune response tuning, so for antimicrobial peptides, reactive oxygen species, and so it can lead to different strategies of tolerance of the symbionts or resistance. Another control mechanism is the metabolic delivery, because the host can deliver specific metabolites and impact the proliferation rate of certain symbionts or sanction the ones that are not doing the job. And also tissue specificity could play an important role, because like in a squid, where you have the symbionts in the light organ, there are other systems, especially in insects, where you have bacteriums that harbor symbionts, and the fact that they are within specific organ allows a specific regulation within this organ in terms of immunity and different regulation in the organ versus the rest of the body. And in terms of evolution, we can have different type of selections, either the between host selection where the best transmitted strain is selected for, or the within host selection where the most competitive strain is selected for. And so the question that I address in this context is on the regulation of the symbiotic density and in the mechanisms that are involved in tolerance. So if we go back to Drosophila, what is interesting is that in some, you can have different strains of Wolbachia within Drosophila, and one of them, which is called W-male pop, is very interesting in the way that it can be very heterogeneous, which is a rare case in vertically transmitted symbiosis that are generally very homogeneous communities. And this strain, in fact, the variation comes from a region which is called octomom. It's a region of 6 genes that is duplicated in tandem, so you can have different repetition of this region. And what is interesting is that there's a correlation between the number of the octomom region and the density. So if you have a lot of repeats of the octomom region, you will have a higher density than when you have a low number of repeats. And so you can have within host heterogeneity and between host heterogeneity. So it makes a great tool to study the role of the different selective forces that can be involved in this system. So what we did is, first we look at the variation of octomom number depending on the age of flies. And what you can see here is that this number increases with age, which means that it suggests that there is within host selection in this system. And something that generally is not taken into account in vertically transmitted symbiosis. And then we took flies from different genotypes that had different compositions, some with a high number of bacteria and some with very low number of bacteria. We made integrations, and what we could see is to determine who is responsible for the regulation. And we could see that the host genotype is not involved in the regulation, but it's more the initial bacterial pool that plays a role in this density regulation. And when we let the line evolve over time, we have a lot of variations that may be attributable to drift. So in these systems, drift seems to be a very important point, because there is a huge bottleneck of transmission of symbionts through the eggs. And so this bottleneck could have a very important role in establishing this drift. Now, if we go in more details in the mechanisms, we will go back to the squid vibrio system because we have many tools in this system, so it's easier to go into details. One question I was asking is, do the symbionts modify the niche, and how they modify it? And so here in this picture, you can see on the left side, it's a light organ of an aposymbiotic squid 2 days after emergence. So you can still see the appendages with the cilia on the left. And if you look at the right, it's the same organ, but which has been colonized by the wild-type Vibrio fischeri. So you can see that there is no more appendages, it's very reduced, and you have a degenerescence of the tissues. And in the middle, you have an organ that has been colonized by a mutant that doesn't produce light, delta lux mutant. And in this case, you have a kind of intermediate phenotype between the aposymbiotic and the wild type. And Eric had made an interesting observation, is that it's possible to recolonize with bacteria an aposymbiotic light organ. So even if it's 2 weeks after emergence, if you put Vibrio fischeri, the squid can still be colonized. If you do that with a squid that was initially colonized by a mutant, you can again colonize with a wild-type fischeri. But if you try to colonize a squid that were initially colonized with fischeri, but where you have removed the Vibrio fischeri in the first 2 days, so it's again an aposymbiotic squid, but he has experienced initial colonization event, then you cannot recolonize it, which means that there are processes that block a recolonization system. And so these results and others from Margaret McFolney and Ned Ruby's lab showed that there's an irreversible phenotype, which is induced by the first contact with Vibrio fischeri, and which is linked to the morphogenesis of the light organ, and the fact that the appendages regress and there is no cilia anymore blocks the new colonization. And so as soon as you will have contact with a wild-type Vibrio fischeri, all this irreversible process will occur. And what I was interested in is to look after this regression occurred at 4 weeks, or when the squid is mature, and compare the gene expression in these 3 categories. And what we could see is that when you look at 4 weeks, so there's no more delta lux because they've been eliminated, or at least they are lost from the squid. And they really look like the aposymbiotic and not like the wild type, even if in terms of structure, they look more like the wild type. And this shows that, in fact, there's an accommodation of the presence of symbionts. So as soon as you have symbionts in the light organ, you have specific gene expression, especially in terms of metabolic adjustment, chemical homeostasis, and this is really linked to the presence of symbionts. As soon as you remove the symbiont, you don't have this signal anymore. And it's even more complicated in a way that this system is rhythmic, in a way that if you look at the day-night cycle, every day at dawn, most of the bacterial population is expelled from the light organ, and the 5% remaining will grow again the next day and reach a plateau at the end of the day. And so every day this cycle starts again. And so we were wondering if the genes that we showed that were linked to the presence of symbiont will change their expression along that rhythm. And it's what we observed. In fact, when the symbionts are released, the signature in terms of gene expression is very similar from aposymbiotic and symbiotic squid. And as soon as you have symbionts in the squid, like at the last 2 points on the right, you have a strong differential signature. So to summarize this part, I showed you that there can be heterogeneity within the host. So this is well described in horizontally transmitted symbiosis, but it was not so much taken into account in vertically transmitted symbiosis. And so this opens new questions on the source of heterogeneity and the potential for rapid evolution in this system, even though it's challenging in terms of follow-up because we need to find proxy to follow this evolution. It's important to look at the variation of density over the course of the individual's life, but even within a day, I would say. And we showed in this example that the host can adjust and control tolerance mechanisms according to the density. And it's important to look at different tissues because depending on the tissue, more or less symbionts will be present and you can have different regulations. And finally, drift seems to be an important player, especially in vertically transmitted symbiosis. And it could limit the efficiency of selective process. And so what would be interesting if we would like to continue on this aspect is to quantify the intensity of bottlenecks and the impact on host adaptation. Now I will talk about multiple interactions. If you're in an ecosystem, you have several players that can interact with each other. And so if you have just one symbiont, you have one extended phenotype. If you have several symbionts, you can have several extended phenotype. And you can even have more than 3 different extended phenotypes because they can combine or inhibit each other. And so these multiple interactions are important to be taken into account. So one example is a nutritional symbiosis with multiple partners, where different symbionts can produce different metabolites that are so they can use different metabolites that are used by the host. And so you can have a system where 2 different symbionts provide the metabolites or 2 different symbionts cross-feed, and its outcome will be a metabolite by one of them, but it requires the presence of other symbionts. Another example is a protective and defensive symbiosis, where you have a mutualistic symbiont that inhibits the growth of pathogen. And this can be due to competition for resources, production of toxins, priming of the host immune system, or improvement of the host vigor. And so what I was interested in this part is to characterize the mechanisms of protective symbiosis and to see if we could have a combination of nutritional and protective symbiosis. So to do that, I worked on fruit flies again with Wolbachia, infected by Wolbachia. And the pathogen I use is a Drosophila C virus, which is a common pathogen of Drosophila, natural pathogen of Drosophila. And what is known is when flies are infected by DCV, there is an obstruction of the digestive tract, as you can see here with the increase of the crop, and it leads to the death of flies. And so what is interesting in the system is it has been shown that Wolbachia can improve adult survival after viral pricking. And it's a thing that is now used in control strategy in mosquitoes to fight against transmission of viral arboviruses. And so here, what we did is we look at the impact of DCV on fruit flies, but in natural condition of infection, so through ingestion and not pricking, and considering the whole life cycle, and to try to see if we could extract some protection, symbiotic-mediated protection mechanism. So we did the infection through feeding, as I told you, and we could see that the quantity of DCV of the virus decreases when Wolbachia is present. So there is pathogen blocking as for pricking. And then we look at the life history tree of the host, and we could see that if you look at the graph on the right side, the survival curve of the adult, and you can see that the ones that don't carry Wolbachia die much faster. So there is a protection of Wolbachia in the adult stage. But if you look at the left panel in the middle, you can see that there is no difference in terms of carrying Wolbachia or not in the egg to adult survival rate, which is intermediate development. And so we don't see any protection in large. And then we did some transcriptomic and metabolomics, and we found very, very few genes that were differentially expressed in guts. And there are many related in immunity and in metabolism, but it's not easy to make the proper link. There are very few metabolites that are differentially represented in flies. So they are linked to amino acids, T-cell cycle, and lipids. And surprisingly, there is no direct impact of Wolbachia on this signature, which was a bit disappointed. So we continued this project and complexified it a little bit in a way that we added the gut commensal into the equation. And so Lactobacillus plantarum and Lactobacter pomorum. And the idea was to see if there was a direct impact of this gut commensal on the pathogen, on blocking of the pathogen, or an indirect impact via the host vigo in terms of metabolism or host priming, or finally indirect impact on Wolbachia that would impact the blocking of pathogen. So again, we did that with Vincent Raquin and François Lelier. We used biotic flies that we combine with all combination of bacteria, viruses, and Wolbachia. And what we could see is that there's a positive effect of the combination of both commensal and Wolbachia on fly survival. But what was interesting is that there's no decrease in DCV titer and no change in Wolbachia density. So it doesn't seem that it's a real blocking. It's more due to a higher larval tolerance length. And this suggests that it's a better tolerance to DCV, which is due to a higher host vigo. And in terms of genes that are involved, we found a few genes in immune and metabolic genes, related genes. So to discuss a bit about this part, what we show was very surprising is that even if we had very strong pathogenic effects linked to the death of bacteria, of flies in response to viruses, we didn't see a strong transcriptomic and metabolic response. So we question about this weak signal, and maybe it's just because it's dynamic response and we didn't look at the right time point, or there is very specific tissue response and if the gut was not the right tissue to look at. But what we could see at least is that there's an importance of non-canonical, what I call non-canonical immunity, which can be just due to host vigo or competition for nutrients, and not the classical IMD or TOL pathways that are generally used to fight pathogens. And what I learned from that as well is that study of multiparty interactions is complex. And so we need for realistic simplified systems with more than 2 players, but with genetic tools. And so it's not that easy to establish. And on top of that, using multi-omics analysis with transcriptomics and metabolomics, it's kind of hard to put everything together to have an integrated view of what happens. Okay, so the last point I wanted to talk today is about the impact of stressors. So what I call by stressor is a factor that is able to disrupt an homeostasis. And so you can have biotic stressors, such as pathogen or parasites. But you can also have abiotic stressors, such as temperature, equality, nutrients, toxicants. And what we wanted to see is that if there is a direct impact of these stressors on the host physiology and immunity, and on the symbiont density and competition. But also it could have an indirect impact, so either on the host via symbionts or on symbiont via the host niche modification. And so in response to this impacts, you can have two ways for the association. Either you have adaptation, and which can be genetic or a plastic adaptation, and then you will have a resilience and maintenance of the symbiosis. Or you have no adaptation, and then the symbiosis can break down. And so the question I wanted to address is there a positive in terms of buffering, the presence of symbiosis buffers the stress, or negative, the presence of symbiosis make the system much more vulnerable. And another way to say it is symbiosis favors or limits adaptation to stress. So I mainly focus on oxidative stress. So oxidative environment is an exogenous exposure to oxidizing compounds and the chemical reaction in the cell. And when you have more oxidants than antioxidants, then we call this oxidative stress. And this oxidative stress can have impact on the cell. It can induce cellular damages, have impact on physiology, immunity, development. And so it's important stress to take into account. And so with David Mona and Alexis Bena, we looked at the impact of this oxidative stress that we use PowerPoint to induce oxidative stress on the fruit fly Wolbachia system. And what David could show is that if you look on the graph on the lower part, you can see that the density of Wolbachia is much lower when you have paraquat, so when there's oxidative stress, compared to control or when there's glutathione. So there is a strong reduction in Wolbachia density in response to this stress. And we look in terms of transcriptomic and metabolomics, and we could see that, not surprisingly, there's a large impact on chemical homeostasis and detoxification. But more interestingly, it seems that this oxidative stress seems to reprogram the general machinery and metabolism of the fly. And in particular, it increases the lipid metabolism and it decreases carbohydrate, and especially in interaction with Wolbachia. But what is interesting in nature is that in general, stress doesn't occur once, it can occur several times, and it can occur with another stress. And so you can have combination of stressors. And it's what we wanted to address in this project to determine what is the response to symbiotic association with multiple stressors at the same time. Does evolution, the first stress, impact the evolution to another stress? And is, again, symbiosis driver of adaptation in this case. So what we did is we took flies from different populations with or without Wolbachia, and the Wolbachia they could have variation in terms of optimum repeats. And we apply different stresses. So in black, you have no stress. In red, you have stress only with oxidative stress. In green, stress with only virus, so the DCB virus. And in purple, the combination of oxidative stress and virus. And what we could see, we could confirm the previous results that in presence of paraquat, you have a decrease in bacterial density. in presence of virus, the presence of Wolbachia played an important role in antiviral resistance. But what was interesting to us is the combination of stressors. And since you had a decrease in protection in Wolbachia density with paraquat, and because the protection is generally linked to the density of Wolbachia, since you decrease the density, you also decrease the protection. And it's what we observed in combination. So we have a system, we have genetic diversity and heritable variation. We have selective pressure that works. So we started experimental evolution experiment. For 12 generation, we applied these stresses for 12 generation. And after 12 generation, we just look at the effect of these stressors on the response to viral infection. But unfortunately, we didn't find anything. Any change in vital density and composition, and no increase in antiviral response. So this was very disappointing and suggests that it's maybe a limited adaptive potential in this system. So to conclude here, we show that the combination of stressors can change the game, and as a symbiotic there's something in here, but so what is important to say is that the symbiosis can impact ecosystems, so it's important to consider relevant combination of stressors when you look at a symbiotic system. Contrary to what is generally thought for horizontally transmitted symbiosis that could play an important role in the rapid evolution, in vertically transmitted symbiosis, the one that I have studied here, it doesn't seem to be the case. And so either it's because they have very limited genetic diversity, and so it's not enough to play a role in rapid evolution, or both can be visible. The drift is too high that it limits bacterial selection. And overall, what I learned from this is that the study of rapid evolution is difficult, and experimental evolution is kind of complicated experiment to set up, and especially because it's difficult to follow the evolutionary dynamic within the host and between hosts, and we need markers of selection and plasticity to address this question. So overall, yeah, I think I will stop here. I hope I have convinced you that the framework of considering a host as an ecosystem with many interactions and different level of selection, it's an interesting scientific framework, because we can address new questions like the role of the environment, microbiota composition. We can look at the community assembly, the ecological dynamics of these communities, address the question of heterogeneity within a host or within a tissue, look at the influence of host evolution that we don't generally do, look at rapid dynamics, evolutionary trajectories. So it's a very nice playground to study the evolution of interactions at different scales. And I will just. I won't have time, but if I have more time later, I can talk to you about it. I will just like to acknowledge all my funding sources and the platform on which I'm working. All the students with who I worked, and especially Vincent Raquin, David Monin, Alexis Bénard, Marie Poulin, and Raphaël Georges, all the people in the BBE that work with me, and different collaborations have established in this work. And thank you for your attention. Do you have any questions?
Yes.
No.
Okay. So if you have questions.
I think among the many questions of this last talk, there is one I think which maybe is quite interesting, is that when you've got a population of hosts and associates, symbionts, the population structures, genetic structures, the spatial scale that everything is happening is different for the symbionts and the host. And therefore, probably you cannot have adaptation of the symbiont to the host in some cases.
Yeah, it could be.
And so manipulation by the host can be relatively easy because of this lack of adaptation.
I think it depends on which type of symbiont you have. If it's vertically transmitted, you may have adaptation more easily than if it's horizontally transmitted.
Vertical also is very interesting because there's a tendency to say, well, it's vertical transmission, so the interest of the symbiont and the host are aligned.
Mostly, not
mostly. But I mean, there are very nice experiments showing, for instance, that there is within cytoplasm selection for functional mitochondrial circle.
Exactly.
So, in fact, what is happening is that you've got a habitat for the symbiont, and this habitat, which is shaped by the host, is what selects the symbiont.
Yeah, definitely. And we can have conflicts with mitochondria. All these nucleocytoplasmic incompatibilities can occur, even if it's vertically transmitted. It's not only, and especially when it's transmission only with females, with Wolbachia, we have the case in terms of reproductive manipulation. It's a vertically transmitted symbiosis, but you still have a lot of conflicts in it. So it's kind of general patterns, but it's not the only pattern. And I agree that it's the cell itself which makes the environment in this system.
I think the concept of individual selection happening on the symbionts, on the associates, is within the host is very interesting. Yeah, I agree. Thank you.
So this question of having a lot of drift in vertically symbionts come. I mean, it's a recurrent theme even for mitochondrial evolution. So I was curious to see that you were saying that in the case, I don't remember the gene that had multiple copies in the back of genome that you mentioned. Was all about drift in your experiment. I didn't get exactly how you came to that conclusion, and I was also wondering whether there is recombination in Wolbachia genome, but that's a side point.
So in terms of initially we were thinking that it was a host determinism because I did interrogation in different genetic background, and I had a lot of variation depending on the region from which they were originating. So I had different population with different levels. I was thinking all grade the host impact, the impact of the host genotype, and in fact, over time it kind of faded already. And then when I did again this integration, I had very different patterns. So it was definitely not the host genotypes that was in play. And that the reason why we made this clean integration, and when we suspected the drift to have a strong impact, and with time, everything was going in all directions. And so there was definitely not a strong selection for high or low copies.
But the gene was lost in this case, for instance?
It was not lost, and the problem with the system is we can only measure a mean optimum number, and so within the host you have diversity, you have Wolbachia that have very low number and Wolbachia that have high numbers, and we cannot separate them. So it's just a mean number. So we don't have exactly an idea about the heterogeneity within the host. We just see this mean changing over time. So I cannot say that it's lost. It's not lost in some bacteria. It's just that overall in the population it is not lost. But we with Mathieu Sicard, he just submitted a project on these questions about rapid evolution and how this octagon gene duplicates or is lost, and all this recombination we could see in these genomes. So it's something we are addressing right now.
So maybe a question related to your last experiments. So you had, I was wondering about the level of genetic diversity in the lines and whether they differ in Wolbachia titer and in virus protection.
So in terms of it's related to
exactly.
this question, so it's the line we used. So we integrate them within a W. So we integrate a WL population in the different strains, and so we have variation in terms of number of Wolbachia, number of octomom, mean number of octomom, and protective effect. So it was well correlated. The one that had a lot of octomom and a lot of Wolbachia and had a lot of protection.
Okay. So it was you that integrated. It was not okay.
So I enjoy in different. So the background, the genetic background, is used from files we collected all around the world. But we introduce our WMAPOP in these backgrounds.
And so the variation in titer you see among lines, do you think it's because of drift mostly and not because of the host background? Is it your conclusion?
Yeah.
Okay.
Yeah, we were kind of surprised. So we did a lot of experiments. Drift in vertically transmitted symbiosis is not surprising, but it was surprising that it was so strong. In terms of effect, we would have expected that since the presence of octomom has an impact on density, you can have easily an impact on life history, threatening and survival when you rear them at high temperature. And so we were expecting to be able to select line with high number and line with low number, forcing with temperature or with rapid reproduction.
Yeah, thank you. Just more conceptually, this idea of the host as an ecosystem. I was trying to think, like you talk about heterogeneity, for instance, within the host, but I was trying to think what is actually the ecosystem? Is it the whole host, the whole Drosophila, the whole squid, or you have separate ecosystems within the host? So like the system around the optical thing is different than the gut, is different than another tissue. So it's my question is like, what exactly do we mean with the host as an ecosystem? And then as a more generic, like, what does it brings us more if we think it as an ecosystem or if we think it as interaction between the host and this more smaller scale bacterial community within the host?
Okay, so in terms of ecosystem, I think it's a nested ecosystem in a way that you have the big ecosystem, which is the host inside, or the squid or the fly, and you have nested ecosystems that are in organs. And so obviously what happens in the ovaries versus in the light organ, in the bacteriums can be different with different regulation. And it's like you have small ecosystems within a bigger ecosystem. That's the response to the first question. The second question, I think it's quite close to consider them separately, and just to, in terms of how we, I'm sorry, I was 6 week, it's more in terms of which tools we could use, and thinking it as an ecosystem, to me, helps to bring some tools from a community ecology and things like that we wouldn't necessarily think about if we studied them separately.
I was wondering about the multiple stressors. So when did you apply either stressor combination of either is together at the same time, was pre-selected or pre-exposure and exposure, and also when the infection occurred, was already infected Drosophila and then you applied the stressor, or yeah.
So what we did is we applied the stress during the larval stage. So it was in the food. So they were continuously with the stressors in terms of paraquat and viruses. They were eating this stressor all the time. So during all the larval stage, then at the adult stage, they were moved to a free, stressor-free diet. And so we did that every generation. So when the stressors were together, it was exactly at the same period of time. It was not one and then the other. But we did a separate time scale when we did the experiment, for instance, in evolution with paraquat during 12 generation. And after 12 generation, we look at the impact of viruses on the lines that are involved with paraquat for 12 generations. So in that case, it was a separate stress.
Yes, and I have a question for the same experiment. In Drosophila monogaster, there are two strains of Wolbachia, like 3, but and you use for corn, but in nature and WML and WMLCS, and they have different strengths of influence on phenotype on protection phenotype. And which strain did you use for your experiments?
So for the experiment, I didn't use natural strain because it's not strong enough. So there's a variation, but WMLCS has a higher density, and so it's a little higher protection, but it's not very strong. And I needed a strong protection to apply a more important selective force. So I used WML pod strain, which has a more efficient protective effect. But there was variation in terms of number of copies. So there were some Wolbachia that carry very low number of octomom and others that have higher. And that's the reason I wanted to have this heterogeneity to be able to select for strains that have more. I was expecting to have an increase in the number of octomom repeats over time when I stress them at each generation with the viruses. But no.
Yes, and I have a technical curiosity. You mentioned at some point, I think, that you can track you will need more temporal to understand something. So how can you technically track the abundances of the bacteria in a Drosophila?
So the density is you need to kill the fly, but it's very easy with quantitative long-time PCR.
Yeah, but then you killed it, so.
You kill it.
So you cannot track it anymore. Then you have to track another.
So you let them lay their eggs and then you kill them. So it's you let them, you put them in small boxes where they can lay their eggs, and you use these eggs for the rest of the experiments, and you take the film mass and you make an estimation of the quantity in the female that have already laid their eggs. So it's not a problem.
And the squid?
Oh, in the squid is more complicated. In the squid,
Because I thought something that has a longer. I didn't think that we talk about them through generations, because I thought that's an individual.
oh no,
So I don't think that's
different. It's yeah, for flies, each generation you
okay.
and so.
But the squid?
But the squid, we didn't do experimental evolution because it's not, you need to kill the squid. So at that point, and it's already complicated to make them lay their eggs and to raise them in the lab. So far it's very complicated to make to do experimental evolution on squid. You can do a lot of things on dissecting the mechanisms of interaction in the system, but all the evolutionary part, we need to have better system management.
So maybe a question about the squid system as well. From the Vibrio perspective, like do they get something out of it, like nutrients or stuff like that? Or is it just like being in the water, but they're in the light organ?
So it's better. It depends in a way that in the seawater there are very low concentration of Vibrio fish, right? compared to in the light organ where they can develop at very high density. And the fact that they are vented every day in the seawater, it's kind of seedling of the surrounding seawater. And so the baby, when they hatch, they are surrounded by bacteria that have been expelled by their mother. So it could be a kind of transmission advantage.
Like pseudo-vertical transmission. So do you see it, do you see genetic differentiation between the Vibrio around squid versus some random sample in the ocean? Like, do you see?
So in the ocean, in the middle of the ocean, you have almost no fishery if you collect water.
But where you don't have squid, like if
but
you
yeah,
don't have
net
population
rapid.
of squid, like could you?
Net rapid, it's an experiment, and you have different strains. In fact, they are from Hawaii, and the current is going this way, and so you have two populations of squid that are slightly different in terms of morphology, and they don't have exactly the same symbionts. And so this would go into your direction, but that is kind of specific.
And can you swap the symbionts?
We can. It works, but some are. It depends on the strains. The work from Petit Bongrand, she did a lot of swapping experiments to see which one are the most competitive ones. But you can do genetic populations studies on this.
There's no more question. I think we're going to just thank you once again.